should we wait for a few minutes? Like, shall we give it like 2 or 3 minutes?
Yeah, sounds good.
Because people are still joining coming in, so. I think the important thing is, apart from the people joining in, because it's now available on our YouTube channel, we have people viewing it later. And, you know, some of the people who also log in would want to listen to it later if they've, you know, not understood it, because you can, you know, replay, pause, and do all those functions. So it's really fantastic that we have it on our YouTube channel. And so I will just start off until people join. So today is our last seminar for our seminar series.
Just it's the one before the last.
One before the last. Okay, I've got it completely wrong. Today is the third one, is it?
The fourth one.
A fourth one. Okay, okay. Yes. Sorry. Got it all wrong. Got it terribly wrong. So today is the fourth one, but we've got another one. So in our seminar series, welcome all of you for the fourth one. And I think it's been a very successful seminar series so far from all the feedback we are getting. And also, when you look at the number of views of the ones that have been posted on YouTube, there are quite a large number of views, which is obviously very promising and just indicates the need for such a webinar series. And we've got a huge response and a huge interest for this webinar series. So would really like to thank Dinithi for this. So who is one of our speakers? It's Dinithi who put everything together, got their whole team organized to actually put forward this seminar series. And it was just very randomly that me and Dinithi met, which was, you know, like coincidence. When Dinithi emailed me about, you know, doing some work together, I happened to be exactly where she was sending the email from. And so that, I think that's quite a coincidence, and this was meant to happen. But anyway, since we have a forum now, let's start off. So we have two speakers now. I will introduce both speakers. I mean, the second speaker needs no introduction, but still I will introduce. The second speaker is Dinithi, but I'll introduce the first speaker first. And I'm really sorry if I get the name wrong. Which is Dr. Rasa Elmentaite.
Elmentaite, that's all right.
Okay, I'm so sorry about that. But just to introduce Dr. Rasa Elmentaite, she's a staff scientist at the Wellcome Sanger Institute. She's a fellow with Company Creation and Venture Group Foresight Capital. She has a PhD with Sarah Deckmine at the Wellcome Sanger Institute, University of Cambridge. She has a BSc and MSc in Molecular and Cellular Biology with Biotechnology from University of Glasgow. So doctoral research at the Wellcome Sanger Institute, Rasa has focused on building human gut cell atlas using state-of-the-art computational single-cell analysis approaches. In her latest work, first author publication in Nature. Wow. Okay, Rasa, I mean, that's so impressive. She detected the cellular mechanisms dictating gastrointestinal immune tissue development, highlighting the adaptation as a key event in pathogenesis of Crohn's disease. Rasa is interested in applying single-cell sequencing technologies and in silico methods for drug target prioritization and validation. Currently, she is spearheading the translational and her human cell atlas research into a startup company on self-focused drug target discovery. Wow, that's just amazing! Such an inspiring, you know, story about, you know, being such a brilliant scientist and then working towards a startup company. And I think that's the future. And then, of course, coming to Dinithi, again, I would really want to thank Dinithi because Dinithi is instrumental in making all this happen. So thank you so much, Dinithi. So Dinithi joined the TechMind Lab 2 years ago, back as a Marie Curie postdoctoral fellow to focus on computational and machine learning models for comparing in vitro and in vivo single-cell data. She's a computer science and engineering graduate from Monash University and obtained her PhD in computational biology from Monash University, Australia. So we have a wonderful Dinithi spearheading everything, speaking today. So over to you, Rasa, and then Dinithi. So thank you so much.
Thank you for the introduction. I hope you can hear me well. And I also want to thank Dinithi for inviting me to give this presentation today. So from my introduction, I am a staff scientist currently in the Teichmann lab. And the focus really of my position is to understand ways how we can use human cell atlas data that we generate and the tools that we create in translational science. So for drug target discovery or cell engineering. And this is a position that I've been doing for the last year and a half. And part of that process has been to create a company out of these ideas. So today, the first part, I'll be talking about some of the concepts in translational science and some of my work in the gut and how that relates to translational science. And then I'll hand over to Dinithi to talk about cell engineering and her tools that I find super exciting as well and use them all the time. I want to start by kind of going back to the vision and mission of Human Cell Atlas, which is of course to resolve the cell heterogeneity of all the human tissues. And while it's hugely complex, the heterogeneity of these cells, we can broadly categorize them into two functional groups. And that kind of helps to comprehend all this amount of information. So the first group would be the primary cells. These are the cell types that perform very generalized organ-specific functions, and they tend to be quite distinct for each different tissues. So these cell types include neural cells in the brain, myocytes in skeletal versus cardiac muscle, or epithelial cells that we find in lung and the gut. So while there are some cell types that, epithelial cells that are both in different tissues, they tend to be quite distinct from each other, both on transcriptional profiles and their functions. So that's one category. And the second category would be these supportive cell types that are present across different tissues, and they generally support the main function of primary cells. So we can think of, for example, fibroblasts that are specific to intestinal tissues, and they secrete factors to support the proliferation and differentiation of epithelial cells in the gut. And while we profile all this huge heterogeneity of cells, we can start making connections of how these different subtypes are very more similar to each other across different organs, and how they're distinct, and what kind of functions these cell types perform. So of course, this is quite a fundamental task, but at the end, what we want to get to is this comprehensive reference map that we can then use to understand and diagnose disease. How can we envision that's gonna work? Well, we can envision that the reference Human Cell Atlas is gonna be this kind of a UMAP atlas, where once we profile more disease samples, we can overlay these cells on top of our reference and identify the cells that are out of reference. So for example, cancer cells, we would imagine would really distinctly separate and would not be something that is present in the healthy tissues. But in a lot of different diseases, this is much more complicated pictures than in cancers. And the information that we can learn from such kind of understanding of which cell types are out of reference type of cells, we can better understand their expression programs, how they develop, how they differentiate, what type of origin they have, and how they interact with their environment. So that's kind of really the goal, to first generate this reference atlas that we would be able to then use to better understand the disease mechanisms of the cells that are perhaps either unique to disease or change substantially in disease tissue. And throughout my time as a staff scientist, I've been trying to think of different ways of how can we enable translation using human cell atlas. And I think having disease tissue is, of course, something that we all want to have to directly look at the tissue, but there's a lot of things that we can learn just looking at fundamental tissue architecture and the mechanisms of different tissues and how the cells operate in that tissue. But generally, you could kind of categorize it into three categories. The first thing is the fundamental tissue understanding that perhaps can inform what kind of drugs would affect on target but off tissue, and identify some clinically relevant biology of specific tissues. And I'm going to talk about this today. The second part is you can identify how the cells change directly in tissue, so by profiling disease samples. So this is kind of an example where you overlay your disease samples on the healthy reference. And then the third part is how you can model the disease. So that's actually really related to Dinithi's talk and her compartment, where you can better understand how the in vitro models recapitulate the healthy tissues, and how can we make these models better and perhaps engineer cell types in a dish. To enable all of this translational kind of application, there's been a lot of different computational tools that have been developed, both in our group and elsewhere. And I just wanted to touch on some of these because I will come back to some of them in a specific example. But we can categorize these tools into different tasks as well that all enable this understanding of disease tissue. So for example, we have tools that enable integration using data from different modalities. So for example, if you have single cell data and you would like to incorporate that with your spatial data from spatial transcriptomics, you can use a tool called cell-to location that has been published from our group as well. If you want to identify cell types and their VDJ information, so T cell and B cell receptors that are expressed in immune populations, and infer a trajectory out of this information, you can use a tool called Dandelion. Or for example, if you want to integrate different modalities such as attack RNA and so on, you can use multi-map. And that kind of enables building of these atlases that are diverse in different modalities. The second kind of line of tools would be those that enable large-scale integration and interpretation. And two tools that I wanted to highlight are Cell Typist, because it really enables to automated annotation across many data sets, and that's in fact a lot of the work that we do now, trying to harmonize the annotations across different data sets that are being published. And a tool called Milo, you've heard from Emma in your previous sessions, and this is a tool that she developed to better understand which cell types are statistically enriched in specific conditions. So that really helps to identify cell types that are enriched in disease scenarios, for example. And the third category of tools are those that really help to make biological insights into your system. So I've listed a couple of them again. CellPhoneDB is one of the data sets of cell-cell receptor and ligand interactions, which enable to predict from single-cell data how different populations interact in tissue. Drug to cell, which I'm going to talk a bit later about, and genes to genes, which is Dinithi's method that she's going to cover in depth. And so one of the ways that we can really use a human cell atlas to, even without disease data, to interrogate translationally important questions is this tool called Drug to Cell. This bioinformatics tool, it takes Campbell drug target pair database, which has a very comprehensive database of drugs that impact specific targets, and you can retrieve the gene that encodes that target. And so Kazumasa and James, who are the authors of this paper, had this brilliant idea to combine this together with the single cell RNA sequencing data to infer which populations would be affected by specific drugs. So they highly curate and filter this drug data set to filter drugs that have these different categories of drug, clinical trial phase, side effects, and create this sort of matrix of cells by drugs. So which drugs would affect which cell. And from this information, you can not only find drugs that will affect your cell type of interest and kind of prioritize compounds that you would like for a specific disease, but you can also find cells that will respond to this drug. And if you have a comprehensive coverage of your whole human tissues, then that enables you to find potential off-target effects in tissues where you wouldn't expect it. So we, for example, we're looking at some of the compounds that are used for inflammatory bowel diseases, and we could find that some of them had an effect on pregnancy tissues, or had the targets were expressed in placenta tissues in humans. And actually looking through information about that drug that is clinically approved, there are known effects on pregnancies. They're not basically recommended to take for pregnant women. So giving the scale of tools like this and single cell data set, this really enables quick identification of compounds that may affect, may have off tissue effects. But my work so far, from the introduction as well, has really focused mostly on profiling one tissue. I am very fortunate to be in the Teichmann lab, where we have been profiling very many different organs and tissues, some of which are highlighted here in the slide. And some of the understanding of the biological mechanisms that we can target really comes from in-depth understanding of normal tissue. And that's what I would like to talk about today. So for those who don't know what the gut looks like on a cellular level, there's multiple lineages that are present in the normal healthy tissue. And it's a really remarkable tissue in that it changes throughout life and in different anatomical regions, because it constantly has this exposure to the environment. So it has a resident immune system compartment, which starts developing at the earliest times, even before birth. And it becomes more and more mature as we develop during childhood and into adulthood. And it basically interacts with many different other cell populations, including epithelial cells, for example, that line the gut layer. So what we wanted to do in a study that we have done was actually look at more normal development throughout life, including first and second trimester of development. We looked at pediatric patients, both healthy and those with Crohn's disease, and also profiled adults that are organ donors. And what was really unique about the data that we've collected was that we've really tried to cover a lot of different regions of the gut to make sure that this profiling is relevant for the diseases that are most common complex diseases in humans. So these are inflammatory bowel diseases, both Crohn's disease and ulcerative colitis, which are both inflammation of the gut. However, they affect the intestinal tissues very differently. So Crohn's disease manifests in both small and large intestines, especially rectum, and ulcerative colitis has more inflammatory presence in the most distal colon. So what we've done, we've collected samples from multiple regions of the small and large intestines, up to 12 regions actually, from both fetal and adult donors, as well as biopsies from pediatric patients, and uniquely also took some of these samples from mesenteric lymph nodes from both of these categories, with the goal to really create a comprehensive reference of the gut. And this is just a summary of what our reference looked like, and we were able to point out to these cell types that I outlined in the first slide, both that have more primary-like functions and supportive functions. And through analysis of this data, we were able to identify a lot of fundamental or answer a lot of fundamental questions, such as identifying cell types, epithelial subsets that are regionally specialized in one region but not the other. We're able to map Hirschsprung's disease-associated genes, which are which mostly affect the neuronal lineage, this population in brown color, and Hirschsprung's disease manifests in newborns where the babies are born without proper colonization of the distal colon, and so we were able to point out to the populations that might be the ones that are kind of leading to this pathological effect in babies. And the third, perhaps like a surprising line that we've kind of discovery that we found from this data set is focused on formation of lymph nodes and gut-associated lymphoid tissues in humans. And it's a mechanism that happens during development, but also becomes relevant in inflammatory bowel diseases for the recruitment and retention of immune populations. So what we were able to do was to go in detail into this mechanism for gut-associated lymphoid tissue formation and lymph node formation in humans by looking at what has been known so far from mouse development. So we know that these lymphoid aggregates, they start forming early during development, and there are three main cell types that are involved in the formation of these tissues in humans. In mice, and these are lymphoid tissue initiator cells that are ILC3 cells, lymphoid tissue organizer cells that are endothelial in origin, and the mesenchymal cells. These another fibroblastic sort of supportive cell that is called lymphoid tissue organizer cell, MLTO. And through the interactions with all these different cell types, the LTI cells activate the MLTO cells in such a way that they start expressing a lot of chemokines to recruit more immune cells into the developing gut and lymph nodes. So we asked within our data set, can we actually identify some of these populations? So I've told you that we were very fortunate with samples, and we could actually dissect the lymph nodes from human intestinal tissues that appear at around 16 weeks of post-conception. So you can see the white arrows are pointing to some of these lymphoid aggregates that start developing in the bed of vasculature surrounding intestinal tissues. And when we look at the single cell data, we could identify these immune populations that we would expect to see in mice. Only difference is that we actually could see three different populations that seem to have similar characteristics to the ones that have been described in mice. And what we could, while we could visualize this in tissue, all these different populations, we could also see that they're actively recruited to the site where the MLTO cells are in green. And we use these tools such as cell to location to really map in very high precision the collocation of these cells in Visium samples. So this little section here of the developing intestinal gut shows where we find the MLTO cells, which we actually couldn't see by histology alone, but with high throughput transcriptomic methods, we could identify where the cells are and that they co-localize with these other populations that are involved in recruiting more of immune cells into the gut. And why do we think this is important? Well, when we look at the GWAS genes associated with Crohn's disease and ulcerative colitis, we can see that those populations that we identify score really highly in terms of enrichment of genes associated with Crohn's disease. And so our hypothesis from the study was that perhaps this mechanism of immune recruitment and retention during development is quite important in disease as well. And we were able, there's been also studies where people have seen the tertiary lymphoid structures forming in the gut, which look very similar to these lymphoid structures that we see during development, however, a bit more disorganized. And why is this therapeutically important at all? Well, we know that currently for ulcerative colitis as well as Crohn's disease, the drugs that are used and are being developed currently really target the specific mechanism of immune cell recruitment. You can do that by blocking all lymphocyte integrin receptors, and that stops your T cells entering into the gut tissue, or you can actually stop it at a different point where the when the effector T cells migrate from the lymph nodes into the gut tissue, and you can kind of modulate the expression of S1P to really stop the migration of these immune cells into the gut. And so the mechanism that we described during development is a fundamental mechanism of how these cells are organized and recruited into the gut, and could be targetable through different mechanisms to stop the immune recruitment to the gut. So in summary, I delved into a lot of detail of building human gut cell type census across life stages and regions, and how that can inform our fundamental understanding of biological pathways and systems that can be used to inform future therapies as well. And with that, I know that I ran over time, but we have a lot of huge teams of collaborators and past and present team members who've been involved in these projects and have been supporting translational efforts as well. And I want to leave you with this kind of summary of how the HCA can enable translation, and hand over to Dinithi to talk more about the modeling disease using in vitro models. And so if you have any questions now, I would be happy to take any questions before Dinithi starts her presentation. All right, if there's no questions, then Dinithi.
Okay.
Maybe I can share your presentation, or do you need?
I think I will need host permission. Oh.
Hold on, maybe I can do that. How about now?
Yeah, now it works. Thank you.
Awesome.
Okay, so can you see my screen?
Yes.
Okay. Yeah, so thank you, Rasa, for such comprehensive presentation on how the gut studies are being done using single cell data sets. So now I'm going to give you some overview into the computational side of things when it comes to engineering cell-fate in a dish. So as Rasa had shown you, in our lab, there are different organs and tissue types being single cell sequenced to understand their diversity in different cell types and cell states, so that we can understand what's happening in terms of the biology. So if we look at the type of data that we have at hand, this is a typical primary single cell data structure that we are looking at. It's a cell by gene matrix where all the rows are representing cells and all the columns are representing genes. And if you take a spot, it represents the expression, the amount of expression of a particular gene of a particular cell. And when it comes to cell-fate, the cell-fate describes the identity of the cell, and it is usually achieved through cell differentiation process. And we can describe this as a trajectory through a space of different transcriptomic states. So by transcriptomic state, what I meant was that if we take a single cell X like this, the row that corresponds to it is a gene expression vector. And that gives us the transcriptomic state because there are different levels of expression per each gene for this particular cell. And on the right-hand side, you can see sort of a landscape where you could see some parts all the way up to 4 different cell types, which represents mature cell states, A, B, C, D in this case, for example. In here, you could see that it starts from some naive state of pluripotent stem cells all the way up to mature states by following through these routes, different routes to the trajectories. And if you take this cell, you can place this cell on this trajectory according to its transcriptomic state, and that defines how much it has progressed through the differentiation process. So, for example, this is a differentiation trajectory of T cells, which I am going to focus on in the rest of the slides. We start from pluripotent stem cells, and then we will have mature T cells at the end of the differentiation process. And usually this kind of trajectory is explained by a pseudo-time trajectory because we really don't know the absolute time point of a particular cell where it is at the differentiation process. So we kind of can order the cells along this developmental process, which gives us sort of a pseudo ordering. So this is called the pseudo time because of that. And when it comes to engineering, what it means is that outside the body, inside the lab, we are engineering these cell types using artificial engineering protocols. So we start from primary or stem cells, and then based on this desired cell type, we can assign a certain engineering protocol that gives us that cell type at the end. And this includes different growth medium and different factors to add to this sort of process so that it leads us to and commits to the cell lineage and the mature state that we want. So an example of such system is the artificial thymus organoid system. The organoid is an in vitro system, which is actually a self-assembling 3-dimensional tissue construct. The aim of it is to mimic the in vitro cell and gene expression dynamics so that it can actually be used as a proxy or model system to study developmental biology or to study if like responses to different treatments and so on. So it's a very advantageous system where we can which can be applied to regenerative medicine and developmental biology. So here, this protocol was performed, and a thymus organoid was grown by Chen-Chu Suo, who is a clinical PhD student in our lab. So she made this around 9 to 10 weeks to achieve mature T cells. So we were aiming for single positive T cells in here. And the main question we wanted to sort of answer was whether this particular organoid system accurately follows the in vivo dynamics of the thymus T-cell differentiation. So in order to answer that question, we will have some more sub-questions that are a bit challenging. And you can see this as a feedback loop to answer and refine these systems to gain more accurate in vitro systems. So how do we quantify the degree of agreement between these systems? Whether we can pinpoint the differences in gene expression and modules and cell-cell interactions along the time between the in vitro and in vivo system. And if we identify them, whether we can rectify those differences, for example, adding transcription factors to the organoid protocol so that we can refine the protocol. So this is really an iterative feedback loop between wet lab and dry lab side. And there are different learning problems involved. To highlight several, this is about integrating the in vitro and in vivo single cell data sets, and then annotating cell types to understand the cellular composition differences, and then pseudo-time trajectory inference to understand what are the differences along the time, and then cell-cell interactions and perturbation response and so on. And this is where we can benefit from machine learning and AI models today. And there are lots of research being done in this exciting field to address these problems at hand and come up with more refined and more accurate organoid in vitro systems. So for example, in terms of comparison, we were looking at trajectory alignment. So to give you a glimpse of what trajectory alignment is, if you have two pseudo-time developmental trajectories of a reference and a query, so in this case, the reference can be in vivo, and the query can be the organoid. And then we would like to see what are the matching sort of states and mismatching states to understand these systems better. This is a cell level alignment. And then we can also look at individual genes to see how they are aligned as well, to understand what's the sort of differential expression across time as well. So this is an example gene. You can see that the organoid gene has much more upregulated than the reference gene expression. So this alignment, for doing this computational alignment, we developed this new tool called Genes to Genes. It's available as a preprint on bioRxiv if you want to have a look at it. So basically using Genes to Genes, we compared the artificial thymus organoid with the in vivo T-cell differentiation. You can see that there are different cell types as annotated, starting from pluripotent stem cells or going through double negative, double positive stages in T-cell differentiation, all the way up to single positive T-cells and type 1 innate T-cells. So overall, when we are aligning, this is the kind of alignment we are looking at for cell level. And we could see that the time points, these are reference time points, and these are organoid time points. You could see that the early organoid time points are mismatched. That makes lots of sense because the organoid the genes that were different at those last stages were belonging to this TNF-alpha signaling pathway. So we kind of saw this as a potential pathway to target in order to refine the organoid protocol to achieve more mature single positive T cells. So this is some example genes in this pathway. So these are transcription factors. You can see that between the reference and organoid, the reference has much higher expression in here compared to the organoid at the last stages. So these are 4 of the example alignments that we get for each of these genes as well. So with that, what we did was we added TNF-alpha to the artificial organoid protocol, and then tested how much maturity it has achieved for single positive T cells. And then we realized that it has now pushed it to more maturity based on the gene expression distances that we evaluate. So this is a potential target for cell engineering protocol refinement. And by doing these kinds of comparison analysis, we can inform the wet lab side from dry lab side to gain more, to get more accurate systems for more accurate and reliable biological insights. So in summary, there are lots of different protocols for engineering different cell types. Here, what I aimed at was giving an example on T-cell differentiation. And then it is important to evaluate these in vitro protocols to compare with in vitro reference systems to see if it follows the accurate dynamics, and then refine it accordingly. So this is a feedback loop between wet lab and dry lab, and then we can benefit from machine learning and AI models to do this. So with that, I would like to acknowledge all the people involved in this work. This is a really collaborative work between dry lab and wet lab teams. And thank you very much.
So this is actually really interesting because so my work involves T cells, but obviously nothing in depth like this. I'm just a little curious, how long does this roughly take?
Yeah, so this is the protocol. This timeline is actually 10 minutes. Yeah, so
okay, wow.
Yeah, so it takes that amount of time, and then we need to sequence those data and then do the analysis as well. So it takes that kind of time. Yeah.
So if I understand correctly, at each time point, you sort of I mean, you're interacting with your computational team and then sort of modifying it, like you said, with the TNF alpha.
So that's sort of the ideal scenario where we can do the refinement, but it's not very sort of like, so once you have started on a protocol, we have to sort of let it grow, and then at different time points, we can sample and then capture different single cell data sets for those time points. But in order to sort of compare between different refinements, it takes lots of more time than that, because once you have tested one system, one protocol, then you need to do the comparison, and then again you have to start the same protocol. So it takes that much of time. So it's a bit of lengthy process in that case.
Yeah, but you still generate like a lot of really useful data, because what we do is just looking at, say, mature T cells and how they would go about. But this is like looking at the entire thing. So that's really interesting. Thank you so much. I will definitely bug you later on for specific protocols. I think Professor Neelika just, I don't see her logged in because I think she was having some technical difficulties.
I'm unable to actually unshare my screen. I don't know why, but
you are the host, aren't you? Do you see yourself as the host?
I'm not sure. Yeah, I don't see the.
Yeah, I'm wondering if we are having some kind of technical issue, but I think that should be fine.
I see. No worries. Yeah, maybe because we are all already out of time. If there are any more questions, maybe you can sort of send me an email, and I'm happy to answer as well. And
thank you so much. This has been.
Is there any questions in the chat? If there's, I can't see the chat as well.
No, I'm checking the chat now. I don't see any questions posted there.
Okay. No worries. Then. Thank you so much for joining us today.
Okay, thank you so much for organizing this. It's like amazing. Thank you. Okay, then we can probably wrap it up.
Yes.
If no one has questions, I think maybe after people go through the recordings again, they might have questions. So you might have people reaching out later on.
Yeah, sure. So if there's any question for me or Rasa, then please do let us know. And then...
Oh yeah, thank you so much. Thank you, everyone.
Okay, thank you. Bye.
Bye.